Hello everyone. I hope you all are good. In this video we are going to learn about chromosome. In the nucleus of each cell, the DNA molecule is packaged into thread-like structures called chromosomes. A chromosome is a long DNA molecule with part or all of the genetic material of an organism. In most chromosomes, the very long thin DNA fibers are coated with packaging proteins. In eukaryotic cells, the most important of these proteins are the histones. These proteins, aided by chaperone proteins, bind to and condense the DNA molecule to maintain its integrity. These chromosomes display a complex three-dimensional structure, which plays a significant role in transcriptional regulation. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. Now let's see the structure of chromosome. A chromosome has generally 8 parts: centromere or primary constriction or kinetochore, chromatids, chromatin, secondary constriction, telomere, chromomere, chromonome, and matrix. Centromere or kinetochore. It is the primary constriction at the center to which the chromatids or spindle fibers are attached. Its function is to enable movement of the chromosome during the anaphase stage of cell division. Chromatid. During cell division, a chromosome is divided into 2 identical half strands joined by a centromere. A chromatid is each half of the chromosome joined. Each chromatid contains DNA and separates at anaphase to form a separate chromosome. Both chromatids are attached to each other by the centromere. Chromatin. It is a complex of DNA and proteins that forms chromosomes within the nucleus of eukaryotic cells. Nuclear DNA is highly condensed and wrapped around nuclear proteins in order to fit inside the nucleus. In other words, it is not present as free linear strands. The chromatin consists of DNA, RNA, and protein. Secondary constriction. It is generally present for the nucleolar organization. Telomere. Telomere is the terminal region of each side of the chromosome. Arc chromosome has 2. Chromonome. It is a thread-like coiled filamentous structure along which chromomeres are arranged. Chromonome controls the size of the chromosome and it acts as a site of gene bearing. Chromomeres. These are the bead-like structures present on threads or chromonome. These are arranged in a row along the length of chromonome. The number of chromosomes is constant and it is responsible for carrying the genes during cell division to the next generation. Matrix. Pellicle is the membrane surrounding each of the chromosomes. Matrix is the jelly-like substance present inside pellicle. It is formed of non-genetic materials. Now we will see the function of chromosome. The most important function of chromosomes is to carry the basic genetic material, DNA. DNA provides genetic information for various cellular functions. These functions are essential for growth, survival, and reproduction of the organisms. Histones and other proteins cover the chromosomes. These proteins protect it from chemical, e.g., enzymes, and physical forces. Thus, chromosomes also perform the function of protecting the genetic material, DNA, from damage during the process of cell division. During cell division, spindle fibers attached to the centromeres contract and perform an important function. The contraction of centromeres of chromosomes ensures precise distribution of DNA, genetic material, to the daughter nuclei. Chromosomes contain histone and non-histone proteins. These proteins regulate gene action. Cellular molecules that regulate genes work by activating or deactivating these proteins. This activation and deactivation expand or contract the chromosome. Now let's see the types of chromosome. Examples of types of chromosomes. Metacentric chromosomes. Metacentric chromosomes have the centromere present exactly in the center. Both the sections of metacentric chromosomes are therefore of equal length. Example: human chromosome 1 and 3 are metacentric. Submetacentric chromosomes. In submetacentric chromosomes, the centromere is not present exactly at the center. The centromere is slightly offset from the center. Both the sections are therefore not of equal length or are asymmetrical. Example: human chromosomes 4 to 12 are submetacentric. Acrocentric chromosomes. Acrocentric chromosomes have a centromere which is highly offset from the center. Therefore, one of the strands is very long and one very short. Example: human chromosomes 13, 15, 21, and 22 are acrocentric. Telecentric chromosomes. In telecentric chromosomes, the centromere is present at the very end of the chromosome. Telecentric chromosomes are present in species such as mice. Humans do not possess telecentric chromosomes. Diagram representation of types of chromosome. Hey! Don't forget to like and subscribe. Thanks for watching. Keep learning.